What's up, Matrix? It's 6 p.m. on a Wednesday afternoon, so you know what it is. Yes, welcome to Mindset Learn Extra. Matrix, we're going to be doing some life science, some hectic stuff to get you prepared for your prelims. How do you feel, guys? Oh, I'm so excited for you. With me in studios, Adam.
How are you doing, Sia?
How you doing?
I'm doing.
I've been here, so how you doing?
I'm doing.
Okay, he's doing, guys. He's doing. You better also be doing on Facebook. That's facebook.com/Learn Extra. Tweet me at Learn Extra. And guys, if you don't have our notes, I will be posting a link to them, but also you can go get them yourself on our website. That's learn.mindset.co.za, and have fun with this lesson, guys. Remember, the more fun you have, the more you can actually take in. The more you take in, the better your marks are. If you get good marks, you go to good university, you get a good life, you get good money. You know what I'm going with this. So, guys, enjoy the show.
I think I'm going to take you to my classroom tomorrow.
I
need you to tell them that. That same speech. Okay. Well, Mindsetters, welcome back. It's Wednesday again, as Sia said, and we are busy still with our exam revision that we wrote in June. Seems like a very long time ago. Yes, and as Sia said, the prelims are at your doorstep. It also seems like a long time, just like your holiday went very fast. 21 school days left.
What holidays? Matrix don't have holidays.
They don't have holidays. You're right. You are so right. So we are busy with the paper 2 items. Remember this year, unlike previous years, where in the first 2 terms you would probably knock off all your first, your paper 1 stuff, and then you would do paper 2. Now, the way the syllabus is structured, you do paper 1 and 2 stuffs in term 1, 2, 3. Well, not 4. By that time, you should be finished. So we've taken out all the paper 2 stuff now for them, and we're going to give that to them today. So we want you to interact with us and put your questions on Facebook. Remember, we've selected 2 provinces, KZN and Gauteng. If you are in another province and you wrote another paper, please, and if you have a question, take a snapshot, send it to us. We would try and help you answer that question as well. We want you to benefit from this. It's not about me, it's not about Sia, it's about you at home. So you need to be part of this lesson. So let's move on. So what do we want to do? As I said, we are revising the questions in paper 2. Mainly, remember, guys, it's so easy, even for myself. John and I always have a conversation, and he needs to remind me, no, that's paper 1, no, that's paper 2. Because we are used to what we've been doing up until the end of last year. So just be careful. Your paper that you must look at is the Exemplar 2014 to know what is in paper 1 and paper 2. Other than that, the very easy rule of thumb is whatever was in paper 1 last year is now paper 2, and whatever was in paper 2 last year is now paper 1. That's so as simple. It's confusing, but you know you've been doing it the whole year, so it can't be too difficult. The challenge questions for today, and here the reason why I chose this question on genetics, many of you are failing to read properly. And if you can't read properly, you're going to have problems. So I've given you a lot of words in both questions. I want you to look at it very carefully, underline the keywords, and sort it out. It's not difficult. It's just the words that you need to get around, okay? So the question says, if 2 people who are both carriers for a genetically inherited fatal recessive, wow, genetically inherited, so that's something that's passed in the genes, fatal, that's going to kill you, a recessive disease, decide to become parents, what will be the odds? New word, odds, probability.
Chances.
Chances.
Yeah.
Easier word. There you go. What are the chances that their children will also be carriers? So you know what to do there. Remember, they are carriers. They are not sufferers. They are carriers, and the disease is recessive. I leave it there. More than that, I'll be giving you the answer. The next one. A farmer wanted to check the genotypes of black rabbits. He had some black rabbits and some white rabbits. He wanted to know He knows they're black and white because he can see them. Huh? That's phenotype. But he wanted to know the genotype, what's on the inside that we cannot see, the genetic makeup. He allowed the rabbits to mate. The results of the investigation are given in a table below. So we've added a little dimension here. So in the first generation, so he mated black with white, black with white. There you go. Oh, that's rabbits, that's not black. Black, black and white. Huh? So what happened in the F1 generation? All were black. And when he took those black ones, these ones in the F1 generation now, and he mated them again, he found that 75% were black and 25% were white. Now, normally we ask you to give us this probability. Now, we're giving you this probability, and we need you to work backwards, what we call a back test cross. This is what this farmer has done. So you need to figure that one out as well. Now, so far no question there, right?
No, no questions yet.
So where's the question? Let's go back again. Oh yes, okay, there is a question.
They're understanding. They know what's
Right.
happening.
Oh yes, so you must tell us what, somewhere I probably missed out the question in this whole thing. You must tell us what are the genotypes of the original black and white rabbits and the genotype of these two here. Remember what we're looking for? We're looking for the genotypes of the original parents and the genotypes of these two results here that we find. Okay? Good? Let's move on. Remember, you can post your answers on the page, and we will be looking at them from time to time, and we will see where you're going with it. Now we start with the actual questions from the paper. This one here comes from Gauteng. The diagram below shows part of a process that occurs during protein synthesis. So already that process cannot be protein synthesis because it's telling you protein synthesis here. But it's another process within protein synthesis, and you should know by now that there are two parts to protein synthesis for us. Okay? Study the diagram and answer the questions. Good. There's a diagram there. Something is happening here with molecule A, and one of these strands is a coding strand. There comes molecule B. Take careful note at the nitrogenous bases that are found in molecule B, and look at the ones in molecule, if I would call this A, strand A, of molecule A. Good. So they say name the specific process. Now guys, what are the clues given in the diagram? The clue, the biggest clue is this guy here, this shape. Do you remember?
Yes,
What's the shape?
that's helix.
Hey! Wow! Sia is not even studying life science anymore. She did life science when? 3 years ago?
Yes.
3 years ago. Wow. So she knows that's helix, but because there are two, we say double helix. Huh? We say double helix. So double helix is only associated with one molecule, and that is DNA. DNA in protein synthesis is only involved in one of the two processes, and that is transcription. So therefore, this process is transcription. Wow, we got it right. Where in the cell does this process occur? If you know that it's transcription, you should know that transcription takes place in the nucleus. Molecule A and B are both nucleic acids. Tabulate two structural differences between these molecules. So let's go back. We've already identified molecule A as DNA. Obviously, the other one, without even looking at the nitrogenous base, we should know if it's the other nucleic acid. There's only two that you studied, DNA and RNA. So this one is RNA, and there's something in this diagram that already tells you that's RNA. But let's look at our table. Tabulate. So first of all, you had to draw a table. To get 1 mark, you need to draw the table. So that's the 1 mark there. And then you compare. As you compare, you notice you're getting 1 mark on either side. On the table, your comparisons must be next to each other. You can't say here a double-stranded molecule and here it has uracil. Hey? I don't want to go to the boy and girl thing now. We will leave that for later, yeah?
Okay.
When you
Let's
get
leave
used to me.
Yes.
Okay. We don't want to scare you away so
I
quickly.
want to come back.
You will come back, eh? So double-stranded molecule, single-stranded. We can see that there already in the diagram. Secondly, the sugar, but they didn't say visible difference. If they said visible, you cannot use the second one. The sugar is deoxyribose in DNA, and in RNA it's a ribose. Base pairing occurs, no base pairing occurs. Obviously, if it's single strand, it can't be base pairing. Generally, these two will go together, but we're using them there. Thymine is complementary to adenine. Here, uracil is complementary to adenine. Those are the three important differences. We asked for only two, so we've given four there, sorry. And they asked for two structural differences. Reading the question is so important. Not just any, so somebody could have said that one is found only in a nucleus and the other one is found in a nucleus and what have you. But they ask for structural differences, so you stick to that. Describe the role of molecule B in protein synthesis. Molecule B would be the RNA, and that particular one is messenger RNA. What does it do? First of all, messenger RNA copies the code from DNA. Transcription, to transcribe, to copy. So messenger RNA, you're naming it, you're getting a mark for that. It forms a copy of the DNA molecule, makes a copy. It copies the code from DNA molecule. And then it leaves the nucleus. It doesn't stop there. They ask you the role of messenger RNA. The messenger RNA doesn't stop in the nucleus. After it makes the code, I'll relax here. No. It moves out of the nucleus and attaches itself to the ribosome, where it provides the code to the transfer RNA molecule. How? It determines the sequence of the transfer. Actually, I would say determines the sequence of the amino acids eventually. How does it do that? By coding for the right transfer RNA molecule. Remember, the messenger RNA is the codon. In other words, if the messenger RNA is UAC, then the transfer RNA this is providing the code now. So we call it the codon. The transfer RNA that comes to attach to this one must be AUG. And this transfer RNA will have amino acid 1 with it. Another one would have an amino acid 2, and so on and so on. So it determines the sequence of amino acids eventually. The transfer RNAs, really speaking, do not determine the sequence. They simply get the sequence from the messenger RNA. We're still with the same question. Use the table. This is a question that gives lots of you problems of protein synthesis. Use the table below to give the sequence of the 3 amino acids that would be coded for by the section of DNA shown in the diagram above. Now we need to go back to the diagram. So if this is the DNA, this is the messenger RNA. So the original code came from the DNA. It formed a messenger RNA. Now what about transfer RNA? Remember this thing that I've taught you when we did DNA, that the whole of protein synthesis. The DNA and the transfer RNA are the same. The DNA and the transfer RNA are the same. Now what do they have here in this here? Can you see what they have here? Anticodon. Anticodon comes from the transfer RNA. So although they're talking about the DNA, you can't take the code as it is there. You've got to convert that code to transfer RNA code, then read this and get it there. So let's go back. So if that is DNA, this is messenger RNA in the middle of the sandwich. On the other side will be the transfer RNA. In other words, as I told you before, you simply rewrite the DNA first. C, A, we'll write the T, G, G, C, T, A. But we know that in transfer RNA, there is no T. So we have to take out the T there and replace it by U. Take that T out, U. So the first one is CAA. Go down to the table. CAA is this one here. So the first one is glutamine. The second one is UGG. UGG, yes, UGG. UGG, there you go, is tryptophan. And The last one there is CUA. CUA, CUA, CUA is leucine. And let's see. There you go. Glutamine, tryptophan, and leucine. Give the name of the molecule that carries the amino acids in the cytosol. That's easy, the transfer RNA. The bond that forms between two amino acid molecules, that is a peptide bond. Notice the ones that are easy, we're not wasting time with. Is the molecule produced by these three amino acids considered to be a protein? Explain. We can answer this one too. No, because a protein must have at least 50 or more amino acids. Right now, this molecule has 3, so it's called a tripeptide. Sometimes we may even call it a polypeptide. And with that, Sia.
Awesome, Aslam. That is amazing. That's great, actually. Guys, this is all coming back to me just by sitting here. So what's important to note is that if you actually grasp the concepts now, you'll never forget them. Come prelims, you're going to ace those prelims, you're going to ace November. And just to make sure you're on the right track, you can actually go onto our website, learn.mindset.co.za, and actually test yourself. See how many questions you can get right. The more you get right, the more chances you have of winning this amazing Casio calculator. So guys, please do that. But in the meantime, consolidate your notes in the ad break, and I'll see you just now. Welcome back, guys. That was such a short ad break, but I hope it was enough for you to reconsolidate your notes. And don't forget, if you have any questions for me, it's facebook.com/learnextra. We are going to be addressing all your questions, but towards the end of the show. Don't forget, there's a challenge question. Don't forget, test yourself. You win the Casio calculator. Guys, your matrics, your life is busy, but keep up, because this is all in your best interest. So without any further ado, let's carry on with our lesson.
Thanks, yeah. She's a slave driver, guys. I gave you a break so that you can have something to drink. She gave you a break so that you can reconsolidate your notes. Where are you going to fit in a drink in between? I'm sorry, guys. We'll have to reconsider that. We must drink before the show now. Okay, so we're busy with protein synthesis, so we continue in the same line. And we're not going to waste too much of time with the second question. Similar concepts being tested. I went through that one a little slower so that you can see how we get to the final answers. This one, we're going to go a bit slower. The diagram below represents a part of a protein synthesis. This was from KZN, this paper here, this question from KZN. The diagram represents a part of protein synthesis. They want to know, identify the molecules labeled Y and Z. I'm glad that we got these questions, because it helped us to look at both sides of the coin of protein synthesis. But anyway, let's go, and then you'll understand what I'm talking about. So we've got this molecule here. Where is the Y? This one here. Y. And they want Z is this long one here. Okay. So there's one long one there, and there's many of those Y, and this Y is attached to something. Now, we should know by now that that has to be transfer RNA, because it's collecting and bringing amino acids and going away and coming back with other amino acids. Other transfer RNAs are coming. And this one that's here is probably on the ribosome, and it has to be messenger RNA. So this is our labels for those two. One would be transfer RNA Y, and Z will be messenger RNA. Name the phase of protein synthesis represented in this diagram. Phase, we'd rather say stage, but phase is fine. Okay. The one we did in the nucleus just now was transcription. So this has to be the other one, and therefore this would be translation. Transcription is a process where messenger RNA makes a copy of the DNA. Copying. Where is the code? I copy the code, I bring it here for you. And translation, we take that code and we decipher that code to something else. We change the code to something we can understand. What can we understand? We can understand what a protein is. So this was in another language we can't understand. Encrypted stuff, detective stuff. We are doing some forensics. We come there, we decipher the code. We break down this code. We change this code to something more meaningful to the cell. What is this something meaningful? A protein. Why is it meaningful? Because the cell needs proteins. Okay? So that's what we have there. This is translation. Give the name of the group of 3 bases that are indicated by number 4 on the diagram. In other words, this area. What do we call this here? I told you earlier in the last question, so we're not going to waste too much time there. That would be a codon because it's on the messenger RNA. It's providing the code. The transfer RNA is breaking the code, so that's the anticodon. Huh? Good? Write down the base codes from left to right. That would be found at point 3 on the diagram. Point 3 on the diagram is here. And it's matching with this guy. If that is C, that has to be G. Cute girl. Remember? Cute girl. And with the U, it's A. Normally, it should have been at, but there's no T here. So U is A and U is A. So it's GAA. Guanine, adenine, adenine. And again, our friend, the table comes in. Remember, mindsetters, you do not have to memorize anything on this table. If this question is asked, the table is always provided. You need to be able to read. Here's the amino acid here, the name of the amino acids, and here is the base. Listen this one now. This one, they gave you the base triplet from the DNA template. The last question spoke about the anticodon. Anticodon, you must associate to transfer RNA. So here they're telling you, this is what, if a DNA gives you these things here, these are the amino acids. They want to know, write down the names of the amino acids represented by number 1 and number 5. So we've got to go back to the diagram. Number 1 is this one here, and number 5 is this one here. Right, so if that is GUG, that is the transfer RNA. I told you, if the transfer RNA DNA is generally the same, GUG. But if this is DNA, then just like we took out the T in transfer RNA earlier, we have to take out the U here and we have to replace it with a T. We don't put A, we replace it with a T, because T and U are the interchangeable ones, not the ones that combine with each other. That's what you must understand. So GTG is the one, that is number 5, and what's the 1? This is the one here. So this is UGG. Now they gave you the messenger RNA. So the DNA would be the opposite of this because messenger RNA stole the code from DNA. So where the U comes in, now here you'll put A because this is what it would have combined with. Okay, and that's what would have been there as well. So can you see it's the same still? And if that's G, that would have been C, this is also C. If that's G, that's C, this is also C. So your messenger RNA is the opposite of both DNA and transfer RNA. The difference between the DNA and the transfer RNA is the letter U and the letter T. That's what you need to keep in your mind. So we are looking for ACC and GTG. ACC and GTG. ACC, there we go. ACC, that's one there. Threonine, and GTG, valine. There we go. There we go. Okay? We have answered that one successfully. Good. So that's how protein synthesis is taken care of. DNA, if you need to look at DNA again, remember, what can they ask you in DNA? The structure of DNA. They can give you one DNA, you must complete it. They can ask you about the labels of the different things on DNA, the functions of DNA, and DNA replication. And remember, whether they ask you here or they ask you later, you can talk about DNA fingerprinting. They can ask you, it falls under in our syllabus now with genetics, but remember, they're both in the same paper, so it doesn't really matter. So DNA fingerprinting is also important, so you need to know that. And a little bit about GM modification, genetically modified. Again, it falls between DNA and genetics. We're now moving on to questions on meiosis. We're going to try and do both the questions of meiosis here. The following diagrams show chromosomes as they might appear during various stages of meiosis 1 and 2, starting with a cell pair of homologous chromosomes. So they're giving you these diagrams, A, B, C, D, E, F. Give the letters of the drawings that represent meiosis 1. Before we do that, in the correct order in which the phases occur. There's your diagrams. First of all, we need to determine which are meiosis 1 and which are meiosis 2. This one here is definitely meiosis 1. Why? Because we can see crossing over taking place there. This one above, look at the chromosomes. The chromosomes have 2 strands. They have chromatids, both of them together intact still. They haven't separated. So this will also be 1. In this one, look clearly, single strands, chromatids have separated, so this can't be one, so we leave that one out. This one also chromatids have separated and moving apart, so it can't be that one either. Here, it's one chromosome on the equatorial plane on its own, so that also can't be. This one, the chromosomes are lying next to each other, homologous chromosomes, that's also one. Now, the question is, what is the order? And the order would be C first, the chromosomes are lying next to each other, then crossing over D is taking place, and this would be anaphase 1. Anaphase 1, why? Because the chromosomes have separated and they're moving to opposite poles of the cell. So CDA. So two skills here. The first skill you had to know, or to put into practice, was to identify which is meiosis 1 and which is meiosis 2. The second skill was to put those ones that you've identified as meiosis 1 in the correct order. So you have to know your marbles. You can't take chances here. You can't guess here. State one visible difference between the events in meiosis represented in diagrams A and E. That's A, what's happening there is anaphase 1, and this is anaphase 2. By now, without looking at the diagrams, you could have said so. But they ask you specifically only the differences between these two phases as you can see them there. And they are, the whole chromosomes are separating in A, and in E, chromatids are separating. You can't see the centromere splitting. You can't see that there. You cannot even see invagination taking place or a cell plate forming. You can't see that. You can only see what they have there. Good? Name the process in meiosis represented in the phase shown in diagram D. What is the process? Notice they're asking you, not the phase. They want to know the process shown in this diagram here, D. And we spoke about it earlier, so we're not going to waste your time, and that would be crossing over. Notice the difference between the process and the phase. If they ask for the phase, that would be prophase 1. But they did not ask you for that. They asked you for the process that's happening in that phase, and that is crossing over. Understand the difference. Explain why the process named in question 3.3 is biologically significant. Why is crossing over? No matter how they ask this question, what is the significance of crossing over? What is the significance of what you can see in a diagram? The answer is always going to be the same. What does crossing over do? It allows for exchange of genetic material, which brings about variety in organisms. So it introduced, now this one, the answer is put the other way. It's giving you the end first. It introduces genetic variation. Why? Okay, and it's giving you other stories which ensures that the offspring are different to the parents. That's already in genetic variation. I would still give a mark for because it allows for exchange of genetic material. You must add that in your answer, that it allows for exchange of genetic material, which brings about genetic variation in a 3-mark question, and you can add this last part. This can, it may, it's not 100%, but it may increase the chances of surviving because there's a variety available. Good? An organism has 20 chromosomes in its body cells. After meiosis, the gametes produced contain 11 or 9 chromosomes. It had 20. Getting 11 or 9. Explain the events in meiosis 1 that resulted in the gametes having different numbers of chromosomes. That's easy. It's non-disjunction. It means some of the chromosomes did not separate, and therefore one had 11 and the other one had 9. When? During anaphase 1. Or you can say anaphase, where one pair of the chromosome failed to separate, resulting in 11 in the one and 9 in the other. So therefore, the one cell has one more, and the one cell has one less. Remember, you haven't learned this. You've learned trisomy 21, Down syndrome. But it's non-disjunction. So this is an application question of the same topic. You've learned it, this non-disjunction. So you must be able to apply it to a different situation without giving what is this disease. You don't know what this disease is or whatever. You don't know what organism is involved. But you know that it can only happen because of non-disjunction during anaphase of meiosis. And therefore, one has one more and one will have one less. Good? Move to the other question now from Gauteng. The following diagram shows a cell undergoing meiosis. Study the diagram and answer the questions that follow. Here's a beautifully drawn diagram there. Some shading and what have you. And there's only two simple labels, A and B. And this A is the one that you normally get wrong. Because the minute you see A, you start labeling that as nuclear membrane. We'll talk about it just now. Okay, we'll have to talk about it now anyway. Are you with us, Tilsia?
I'm still with you.
You sure?
Yes.
I'm not boring you, am I?
No.
Are you sure?
I'm just getting mad at myself when I'm not getting the answers because I'm like, I know this stuff.
You know the stuff because now I'm going to test you just now.
Oh, God.
You better start Googling there quickly. Provide labels for A and B. So A, I said, can't be the nuclear membrane. So you think you can try that one? A. If it's not the nuclear membrane, what membrane is it?
I'm sorry.
You can't, eh? It's too far back.
No, it's too
Well,
far
if it's
back.
not the nuclear membrane, it's the cell membrane.
Oh,
Remember,
yeah.
guys, remember, Sia, remember, people. When the cell starts dividing, the nuclear membrane disintegrates. In some diagrams, you may see something like this in the early stages where you see the chromatin network visible and so on, and you see this dotted line. That would be the nuclear membrane. But remember, after a while, that's going to disintegrate. It's not going to be there anymore because there's no place for all these other things when the cell is dividing. So you're only seeing nuclear material. All the other organelles have disintegrated and so on. So that has to be the cell membrane. And B is easy. It's the spindle fiber. Does the cell shown above represent a plant cell or an animal cell? First, you have to look at it. Is the plant cell or animal cell? You would say you should have said it's an animal cell. And give one visible reason for your answer. If you look, especially in this area here, you will see that the centrioles have formed the spindle fiber. In a plant cell, the cell wall would have formed the spindle fiber. So that's where our answer comes from, originates. Now, one is that there are centrioles present and only animal cells have centrioles, or there is no cell wall visible and animal cells do not have cell walls, plant cells have cell walls. Remember, when you're giving differences also, try to say, instead of saying it does not have a cell wall, say that this cell has as an outer covering a cell membrane, a positive reason. Whereas plant cells have as their outer covering a cell wall. Try to keep your answers positive rather than negative, unless there's no other way of making it positive. Okay. What phase of meiosis is represented in the diagram? Sia, you think you can manage?
I forgot the phases. It's ana something,
Okay,
is it not?
interphase, prophase, metaphase, anaphase, and telophase. Now
I'd
think
say
of
anaphase.
where these chromosomes are situated. Where are they in the cell?
In the center.
They're in the center or the middle or the equator, right?
Metaphase.
Ah, there you go. You see? A little bit of jogging the memory and it will come back to you. All back. Why? Because you probably understood the work. You did not swat it when you were in school. I'm so happy. I found
fail.
somebody that understands rather than swats. I've been singing that song all day today. Metaphase 2. But why metaphase 2? Because they are in the middle, that's metaphase. But why metaphase 2 and not 1? Because they're lying singly.
Wow.
Don't think that you're seeing two chromosomes, so they're lying together. They're not on the same spindle fiber. They are independently on different spindle fibers. So that means these two are singly there. Their partners would have been next to them, besides them. When they're together, it would be metaphase 1. Now it's metaphase 2. Explain your answer. Well, Kano, you gave them the answer already. Because single or unpaired chromosomes are aligned on the equator. Wow, we've done that one too. We're still busy with that one. Give the diploid number of chromosomes for this cell. Now, how many are here? This is metaphase 2. So there were 2. That means the chromosomes have already separated. So originally, what do we have here? We would have had 4, diploid. Draw the cell in the next phase of meiosis using shading to show genetic variety. So you're showing the next phase, anaphase 2. Why did they say use shading? You need to show these differences there. So they gave you marks for all of these things, for drawing that's the right phase, and for the shading that's done correctly as well. So you know what to do there. Abnormal meiosis can produce abnormal number of chromosomes. For example, Down syndrome, where gamete with an extra chromosome fertilized by normal, and it will have 47 instead of 46. On which chromosome number will there be an extra? We spoke about it just now, chromosome number 21. Explain how this normal was formed in the gamete, similar to what we explained earlier. During oogenesis or spermatogenesis, the chromosomes fail to separate, because they asked you here in the gamete. So you have to talk about that. But remember, meiosis only takes place for gametes as well. State Mendel's principle of segregation, and Mendel's law of segregation states that for each characteristic, there are two factors. And these two factors separate during gamete formation, enabling a gamete to contain only one. So sperm will only have one of the factor, the egg will have the other one. When they come back, the individual will have two. See ya.
Because Aslam said I'm a slave driver, I've got one more task for you. So why don't you go on to www.watergame.co.za and learn more about water, about conserving the environment, and, you know, being a very environmentally friendly person, because if you do that, actually, you have a chance to win 5,000 Rand. Now, who doesn't want that? We have 3,000 Rand for the second prize, 2,000 Rand for the third prize, and up to 20,000 Rand in airtime vouchers. Guys, who are you going to call with 20,000 Rand? I mean, seriously, there's no reason for you not to enter. So go on to the website, that's watergame.co.za, register and play, and then tell us about your experience on Facebook using the hashtag #SAWaterGame. Guys, see you after the ad break. You are allowed to have water now.
On the water.
Hey guys.
I'm out of here, Sia. What did you say before the break? 120,000 for that water game.
Oh, 20,000 Rand for
20,000.
airtime.
I'm out of here. I'm going to go and play the game.
No, afterwards.
After,
no,
okay,
please
okay,
come back.
okay,
you can win
You think I'll
20,000
make it
Rand of
after?
airtime, but if you go afterwards,
Afterwards.
you can't go now.
All
We
right, all
need
right.
you.
Okay, I'll
Guys,
stick around.
before he starts the lesson, actually, let me share something that Bonnie said actually on the page. She says, hey, mindsetters, I hope you guys are working harder and harder since we only have a few months before we write our finals. I wish you all the best. Don't waste a moment, please. And just to add on to that, guys, that's one of your peers motivating you to work harder. I'm glad you guys are motivating each other. But remember, it's also not about hard work. It's about smart work. And that's what Aslam is here to teach us.
Smart is what the whole idea
win
is, guys.
airtime after the
After,
show.
after,
Please
You
stay.
sure? Let me do the smart stuff first. Good, guys. Let's look at this question now. We are doing the last part of paper 2 stuff as we know it now, in what we have done up to now, and that is genetics. Lots of you having lots of problems with genetics, but let's see. The first question is not heavy genetics. Height in humans is a trait that is controlled by many genes. The grade 12s did a certain investigation, and they've given you the results of the investigation in the table below. So they've given you the height and the number of girls. So they only looked at the girls here and their heights, and they've given you a tally 5, 18, 30, 24, 14, 22. Plot a histogram using the information in the table above. We're not going to do much of discussion here as I've done it with the last question where there was a graph. I've discussed the graph in detail. Now we're just going to look at the actual graph there. It's a histogram. Again, you got a heading. I say I'm not going to discuss it, but I will end up discussing it because that's my style. Give you a heading on the y-axis and the x-axis, and notice the histogram, so the bars have to be connected, they have to be against each other. And notice again, the width of all the bars are the same, so you're going to get marks for that. And as you're going this way, on your y-axis, the intervals are equidistant apart. So it's important to have that there. And where there is a unit, you put it in the axis height in centimeters. And that's how you draw that, okay? Now, little bit on investigations. List 3 steps that a grade 12 learner should include in the planning. Not in the method, not in the carrying out, in the planning. planning happens before. You're doing a matric, you're having a matric farewell, everybody must be buzzing now this time of the year. Some people now do it in March and June and what have you. They're not even going to finish the June exams and having a matric farewell. I don't understand the logic.
Freezing, and you're wearing
And
this
it's so
nice
cold, yeah.
dress.
But now, you all want to have the matric farewell. Planning doesn't mean what you're going to do on the night. What you're going to do to get to that stage is planning. And the same goes for scientific methods that you have to do. First of all, look again. We're not doing it. We must decide on the sample size. How big is the sample going to be? Who is the sample going to be? Age groups, etc., etc. Get permission. You're doing something at school, you must get permission. You can't just go out and have an investigation. Even if you're a researcher, if you're doing masters or honors and all that, if you need to go to the school or community, you need permission from the people. Drop a table, survey sheet to record the results. You're going to know how are you going to take the results down when you get it. Obtain the necessary equipment to measure people's height. You can't go there. We're going to measure your height. You look like 1.2. Nah, you look like 1.6. You can't do that. That's not accurate, right? And that's not going to be valid. Arrange a time, a place to measure the girl. So you've got to tell them in advance, this is where we're going to meet. And a venue, we can even add. You must have even a venue, time, or place, sorry. Place is venue, eh? When last we looked. It should be right, yes, yeah? Okay, a
Place
time
or
and a
venue.
place to measure the girl. So the venue. If you see the word venue, don't be confused. It means the place, the area, the classroom, or wherever you're going to do that. Not like me.
They know that.
I got confused with place. Even worse than them. State 2 other possible variables, factors that might have an influence on the height of a person. What else can affect the height? Not only what did they look at here? They looked at number of girls in a grade 12 here, right? Okay, so we must look at other possible variations or factors. Availability and the quality of food most of it is genetics, but obviously with the best of genetics and you're not getting food, it's going to affect your height, you can't grow. Amount of exercise, there is some degree of evidence saying that exercise does influence height, but listen guys, it doesn't mean that I can go and exercise a whole bunch more and I can get tall all of a sudden, taller than I am at the moment. A little taller I would have liked just about 4 centimeters
I'd live
higher.
in the gym.
You think it would happen?
I'd live in the gym.
You'd
if
live in
that
the
could
gym just to get
happen.
that, eh?
Yes.
It happens when you're younger and you do a little bit more exercise, but very, very small difference. So don't get all excited and start exercising. And the production of growth hormones, when do these growth hormones actually start hitting home? That's the other factor that can affect the height. Okay, so that was just more for the graph and for the planning that I put that question in. Let's go here now. Look at where we're going. We're going to something heavy. In humans, the recessive allele, XH, small h here, X, remember, that's the X chromosome, and on it we have the small h, is responsible for hemophilia. The affected individuals bleed easily and what have you, we're not going into that. The dominant allele on the X chromosome ensures that the clotting factors are produced. So it counteracts that hemophilia story. In this case, the individuals are unaffected, do not have hemophilia. The diagram below shows the occurrence of this disease in a family. So there's the story there. We have 1, 2, 3, 4, 5, 6 there. They're giving you the key: affected male, unaffected male, female, affected female, sorry, affected male and unaffected male. So the squares generally in a genetic diagram represent males. My friend Hamida would say because males are square. And
I like it.
females are normally
No.
circles. And what I would say, females are always going around in circles, going nowhere.
Okay, I don't
slowly.
like him anymore.
You don't like me anymore, yeah? You'd like Hamida better, I know. Everybody would say that. Okay, state how many people in this family are affected by the disease. Now they're telling you here. Guys, this is what I don't understand. How can we get this wrong? There's a key that tells you anything that is shaded is affected. Anything that is unshaded is unaffected. So all we have to do is count. You're going to help me here?
Okay.
See, how many are affected if affected is the shaded ones?
3.
3. You sure?
Yes.
You don't want to look behind the screen?
I have my glasses on, but I'm sure.
She's sure it's 3. So let's see. We're going to test Sia now, right? There, it's 3.
Yeah.
So you're right. And they say, and give the genotype. If they are affected, these are all what? According to the schizia, they They're squares, so they are?
They're male.
They're all male. So when we're showing a male, we will always, before even writing whether they're affected or not affected, we always put XY because XY is the male. And XX is the female. We're not worried about that right now in this particular question. But because they are affected, they would have the small h, because to have hemophilia, you have to have the recessive gene. Notice in a male, we do not put anything by the Y, because the Y chromosome doesn't carry any genes. Or rather, it carries very few genes, and one of them is not the hemophilia genes. So that is why more males are, all right, let's not talk about that. Let's see if they asked that just now. What is the percentage chance of individual 1 and 2 producing a child who is an affected male or an unaffected female? And we're not going to go there now. It's 25%, and unaffected female would be 50%. You would have to work it out. Obviously, go back to 1 and 2. 1 and 2. I just want to show you one thing about 1 and 2. This is 1 and that's 2, obviously, right? But notice that one of their children has, or 2 of their children anyway, has hemophilia. So he has the XH factor. He cannot get the XH from the father because the father doesn't have it in the first place. But if he got the X from the father, see, he wouldn't be a he, he would have been a she. Because it's the father who gives the Y. The mother is only XX. She can give X, or X, or X, or X, or X, no Y. So all the guys out there, remember, if you're only getting daughters, don't blame your wife.
It's your fault.
It's your own fault because your Y is lazy. Okay? Let's not go into that right now. But nonetheless, so he can't give the X. So where did they get this X? They had to get it from the mother. But what do we see here? She's normal. So she must have had the normal gene to counteract that one. Can you see how we can work it? So we will have that, and then we will have X, H, Y, and from there you're going to work out a cross, and then you work your answer that way there, right? So I'm not going to go into it. Individual 6 marries an unaffected male. List 3 benefits that could arise if they went for genetic counseling. This is a discussion question, so we're just going to pop the answer. And that, first of all, they'll be able to trace diseases, determine the chances of their offspring being affected, check if the female is a carrier or not, help them make a decision whether to have children or not. Counseled on raising a child with hemophilia. The last one is more important because if the chances are that they're going to get a child with hemophilia, they must know the risk involved, they must know the responsibility that comes with that and the risk of the child dying. So they need to know all that before they make the, remember the counselor does not make the decision. The counselor tells you, these are the pros, these are the cons. You must decide. And this is not only for this, it's for all counseling and for all advice that you seek. The person that's giving advice should tell you, look, these are the benefits, these are the negatives. You must decide. Am I right, Sia?
You decide.
Can
all
I
the
tell you,
time.
Sia, you know, I don't think it's a good idea. You shouldn't do this.
No.
No, I must tell you, look, this is the up, this is the downs, and you must decide.
And I think in all areas of life, it
All
must always
across
be
the
your
board,
choice.
yes. That's the thing. You've got to make the choice. Because when you, even if you make the wrong choice, it must be your choice. Okay? And your fault. Individual 6, right, that 2 children unaffected represent a genetic cross, right? So all we're doing, and here I just want to show you the way that we set it up. Yeah, in this case, I thought I was going to do that. But anyway, individual 6 and the unaffected female had 2 children. Individual 6 and an unaffected female, okay, had 2 children. Now let's see. So if we are saying, if this is the father, or 6, he is affected, so he is XHY. And she is the mother, she will be XH, unaffected at all, not even a carrier, okay? So if meiosis takes place, we will separate that, and I'm not even going to go down here. I'm going to go straight to this one here. These are the gametes. Whatever is on the left, keep it on the left for your own sanity and minds. And whatever is on the right, put it on top. X, capital H, X, capital H, unaffected. Okay? So X, small h, X, capital H for the first one. X, small h, X, capital H. And I'm hoping you're watching to see that no matter what genetic problem you have, the format is the same. Here, the only difference in sex linkage, we use X and Y. And here we will have X capital H, Y, H capital H, Y. So the question wanted to know, just show the genetic type. That's all we had to do here. You could use line diagrams if you like it. It's clumsy sometimes, I find, and you can make mistakes. I would rather you use the Punnett square. It's easier, it's cleaner, there's less chance of you making. But the question says genetic diagram. If you've learned and you're good at a line diagram, you go ahead. But remember about the X and the Y. Whenever you're doing a sex-linked story, you have to use X and Y. And if you're doing something that's not sex-linked, like albinism, etc., there's no X and Y there. Let's go to the challenge question. Challenge question, there, where was it? This one here, okay? And then we'll come back to your questions.
Yay,
all
challenge question.
If two people are both carriers for a genetic inherited fatal recessive disease, they didn't say sex linked. Let's say we use A. They are both carriers, so they must have the small a. But because they are carriers, that means they have the normal gene as well. And that's how you would work out. What will be the odds for the children to be also carriers? That means also like them. And the answer, you would have shown it in a Punnett square, it would be 50%. Remember, 1 is to 2 is to 1. 1 homozygous dominant is to 2 heterozygous. Oops, right? Okay, don't worry about what happened to that circle. But that's what would have happened there. And you would get 50% that way. A farmer wanted to check the genotypes of the black rabbits and so on. So if he checked the genotypes of the first one, he would obviously found that the parent generation, they would have both been homozygous. Homozygous dominant and homozygous recessive. In the next generation, they would have had, in the F2 generation, 25% would be homozygous dominant of this black here. 50% would be heterozygous. There, the first one. And 25% would be homozygous recessive. So it's just going the other way around. That's how we would have worked that one up. I'm hoping. How many of them gave us those answers?
No, actually, most of them, they were arguing about what the answer is, because one would say this and one would say something else, but I think most people are nervous to answer.
They're too nervous, eh?
It's
Guys, you must not be nervous, because even if your answer is wrong, when we discuss it, you can say, that's where I went wrong. This is why my answer is not right. And that's where you will learn from your mistakes. Any other questions, interesting questions that you've picked up?
No, you know, most people answered this question and then they asked work that's not specifically related to what we did in the paper.
Okay.
So I think we'll do that in the in our help desk.
Okay, do that on the help desk, right? Then we'll look at just one question on blood groups quickly. In humans, blood groups are controlled by multiple alleles. How many alleles control blood? It would be 3, because there's A, there's B, and there's O. I wouldn't go to this problem. I want you to work this problem out. Mr. Perro had blood group A, Mrs. Perro had blood group B. They're both heterozygous. Use a genetic diagram to show the possibility of their children's genotypes and phenotypes. I will leave you with that one there. I would leave you with that question. Unfortunately, we've run out of time. And back to Sia.
Thank you so much, Aslam, for that amazing show. Guys, remember, if you still have questions, just post them to our help desk. That's learn.mindset.co.za/helpdesk, and we will be answering all your questions. And then quickly, just a big congratulations to Raymond Nemagovani for winning our Casio calculator for last year.
Raymond.
Yay, go you. We will be contacting you on how to get your calculator. And then from me to you, lots of love. And lastly, a big shout out to my Bali, who's also a life science student and who's going to Norway today. Bye.